Okay, it's 9:03, so I think there's still people joining, but I think we can start in the interest of time. Okay, I'm very happy to welcome everyone once again to the Greener Hub webinar series. I hope you had a great summer holidays. As many of you know, my name is Evelyn Zuniga, and I am one of the communications officers for the Greener Hub project. The Greener Hub project started in 2022 as a coordination and support action of the European Commission under the Horizon Europe program, and it currently represents 15 ERANETs, co-funds, and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs last year in October, and the first topic of our series was circularity in agriculture. Then we moved towards the second series, which was biotechnology to advance sustainable solutions for bioeconomy. So if you're interested in topics related to mixed farming systems, mitigation of greenhouse gas emissions, sustainable farming, production of bioplastics, production of green chemicals, among others, all our recordings are now in YouTube. And now and for these months, we move towards the topic of sustainable food systems, where we have been having talks about saline agriculture, food resilience and climate change, food trade, alternative novel food systems, among many others. I would like just to do a quick announcement for a housekeeping announcement. Feel free to put your question in the Q&A and chat box at any time. I'm going to do my best to address as many questions as I can with the time that we have, which is 1 hour. And if at the end of the session you have a question for either of the speakers, please address the speaker on the question so we know who you're talking about. Today, I'm very happy. We have two panelists, Professor Hervé van der Schuren and Professor Daniel Pleisner. And we are going to open our round of presentation with Professor van der Schuren, who is currently a professor of plant genetics at the University of Liège and tropical horticulture at KU Leuven in Belgium. So allow me just to share my screen. Once again, 1 second. Here. Perfect. Yeah, perfect. Professor van der Schuren has extensive experience in plant science, focusing on enhancing plant resilience and productivity through innovative research and technology transfer. His work includes studying crop responses to biotic and abiotic stresses and developing tools for improving traits in tropical and temperate crops like cassava, banana, rice, potato, and others. His team has developed cassava and rice accessions with increased disease resistance, improved nutrition quality, and prolonged shelf life. He has extensive collaboration with different national and international research projects. And today, the title of his talk is Post-Harvest Losses Mitigation by Improved Plant Healing, which is part of the Feeling project from the FOSC ERANET. Professor van der Schuren, thank you very much for joining us today. I'm very grateful that you want to share the results of all your research with us. I'm just gonna stop sharing my screen and you can start sharing your screen. Thank you very much.
Thank you, Evelyn.
Perfect. Looks good.
Can you see my screen? Full
yes.
screen? Okay.
Yeah, full screen, yeah.
Thank you, Evelyn, for the nice introduction and for the opportunity to talk in this session. Well, you just told me before that we have 15 minutes, so it's going to be quite short, let's say, to discuss about our result, our approach. So we prepared a couple of slides to basically spark some interest into this important topic of post-harvest. So I'll be quite, although I have quite some slide with results, but I think I won't spend too much time in detailing the results, but more trying to do an overview of why it is important to look into plant healing as a potential approach to reduce post-harvest losses. Sorry, I'm trying to change my slides. Okay, so indeed, in our project, we have different objectives. We want to develop new approaches to reduce those post-harvest losses, focusing on plant healing. But we also want to implement novel approaches using, for example, phages to reduce all those important problems with pathogens that occur after harvest. I'm not going to talk about this part of the project, but rather focus on how plants can actually react to wounding so that they can actually, it can also be a mean to reduce post-harvest losses. So if you think about a farmer in the field, he has different options, but very often to reduce losses, food losses, there are not so many options that are actually valid in order to mitigate this problem. You see here 4 different pictures. One is actually, I highlighted in green. This is cassava. It's actually known as a famine crop because this crop can be kept in the soil or in the field for over 2 years without harvest. And this is actually a nice way to preserve or to not to have to care about post-harvest losses. You keep your plant in the field and you harvest as you consume. It's not very efficient for marketing, of course, because if you wish to access markets and sell those crops, those production, this is not very effective. However, for local or for domestic consumption, that can be an option. You also see different other options that you might have encountered when you walk in the fields or in the orchards. You see that for apple, this is not an option. They eventually fall off the tree, and you have to care about how to keep those apple over the winter. If we take the example here, what is happening in temperate climate. You see also other problems of keeping the crop in the fields. You see also sorghum being attacked by birds, or you also see, due to climatic conditions, some germination of some cereals that do occur if you keep it too long. So very often you have to harvest the organs that are produced in order to preserve them and to avoid further losses in the field. So we came into this project with some kind of a focus with our 2 partners. We have a partner in Kenya and a partner in South Africa, where we basically look at different types of problems. As you can see on the next slide, we see here storage of spinach, for example, in South Africa. You see that one way to preserve or to reduce losses is indeed to store them in cold rooms. You've seen directly in the context I showed you before of cassava production, for example, that those kind of facilities are not available to farmers, or not often available. So you need to find also ways to circumvent those limitations that farmers or producers might have in the field. You also see that another example here is tuber crops. You see here the harvest, and actually that's for seed production. So again, good quality needs to, in order to secure good quality and good caliber of the planting material, you also need to remove the rotten potatoes from this production. This is also an important investment, of course, to preserve the quality of the harvest. If you think about cassava, as I mentioned before, a famine crop, for small-scale farmers, they might use it as a food security crop. They keep it in the ground and just harvest when it's necessary. But if you want to empower those farmers and you want them to be able to access some markets, then you need to think about how to harvest this cassava and then transport this cassava roots to the market for selling. And this is where the problem starts, because cassava is, while it's very good to be preserved in the ground, in the field, once you harvest cassava, this is undergoing a decay that is known as a post-harvest physiological deterioration that is actually causing major losses in cassava production. I will get back to this point. So this is what you see here. If you want to compare 2 different crops, if you compare potato and cassava, these are actually 2 different organs. Cassava being a storage root and potato being a tuber. And this makes it also, those 2 organs, quite different in the physiology and the way to respond to damage. You have to see that when we harvest potato, or when we harvest cassava, there's always damage caused. As you can see here on the left-hand side, you're breaking the roots, and therefore you're creating an entry point for oxygen, so for oxidative stress, but also for pathogen. And you see that you can have very quickly some kind of a decay in cassava, while in potato, I will get back to this point, there is a capacity to heal that is relatively efficient, and therefore the losses in potato are clearly reduced or lower than what you observe in cassava. I don't want to go too much into the details, but maybe one thing to highlight here, 2 different contexts. You see here, if you look at sub-Saharan Africa and if you compare with Europe, you see that the places where we are losing food are different locations. You see that It is more during the distribution and the post-harvest that you have major losses in sub-Saharan Africa. You see that in Europe, for example, or other Western countries, you see that there are major losses, or North American countries, for example, you see that at the consumption level, you have major losses. So you see also that the challenge is a bit different. You see that people in places where the value chain, where the cold chain is relatively limited, of course, you have more losses at the post-harvest level, while in places where we are able to preserve the quality of the food and reduce those post-harvest losses, then this is more at the domestic consumption that there is a problem. People do not, or are trashing basically some food, which is another problem, which is of course outside of our focus here, but still very important to be mentioned. So the context is very different, and you see that if you want to increase food security in Africa, for example, targeting this point in the value chain, which is the post-harvest period, is very important because people do not have facilities to reduce, for example, by storing crop production in the cold. And of course, reducing post-harvest losses has many different benefits. As you can see here, you can increase agricultural output. You can also save land if you're producing, or if you reduce the losses, of course, that might also help you to reduce the land that is used for crop production. You also see that the global trend in food loss percentage is relatively stable. So we are still very much struggling to develop solution that helps us to reduce the overall, or the global food losses goes during the post-harvest period. Of course, different challenges. Again, I will not go too much into the details here, but there are different options that can be implemented. Some are implemented already, some are yet to be implemented. If you think especially in the African context, where, as I said before, they don't have necessarily all the different applications. If you think, for example, in terms of coating, because that can be an option, I will get back to this point as well, to preserve or to reduce entry of oxygen. This is something that is relatively expensive and therefore only used when African production or South American production of cassava is actually exported to Northern America or European markets. So we see that there's a price to be paid for that, and this is definitely not a practical solution for local consumption. What I'm showing here are the different entry points for helping this challenge, or at least to address this challenge. You see that we can target different options. And the option we are about to focus on, or we are focusing in the filling project, actually at two different places. One very much focused on plant varieties, so to look at the genetic diversity, how plants respond to post-harvest, how do they keep, or do they have the capacity to respond to wounding, for example, in order to help them to be to extend their shelf life, basically. Packaging can also be an option, as you can see. I will only talk very briefly about this, but I think it's also another interesting application that came out of our research in this project. Okay, so if you look more from the biology point, what you see here on the left-hand side, well, you have two pictures here. This is tomato. I think it's interesting to illustrate also one of the crop we are focusing on, more on the South African component, which is more focusing on preventing losses due to diseases, post-harvest diseases. What you see here on the left-hand side, you see that if you use a dye, you see that shortly after harvest of the tomato, this dye is actually diffusing. It means that at the scar where you collect or where you remove the tomato from the plant, you see that there's a possibility for oxygen or small molecules to actually enter this entry point where you harvest it. You see that after 4 days, the plant has somehow fully recovered, or at least has created some kind of a barrier that prevents oxygen or other small molecules to enter the fruit. That's also very good, of course, to reduce potential oxidative stress, but also to prevent any entry of the pathogen. This is what you see on the right-hand side. You see that after 4 days, the amount or of the potential for infection of pathogens in this tomato is actually seriously diminished or reduced, simply because there is a physical barrier that has been made by the tomato. If you look into potato, well, this is actually a bit more studied as a model system, let's say, for studying healing in plant. You see that also after wounding of a potato tuber, you start to see what we call a wound peridot that's starting to form. Again, I will not go too much into the details, but basically some of those cells that are directly exposed to the environment are basically starting to accumulate some biopolymers that prevents or create a barrier to prevent entry of oxygen, but also entry of pathogens, for example. So that's actually a very interesting mechanism, because if the plant can do that, of course, you have a better shelf life. It means you will have less contamination by pathogen, and you might also reduce other losses due to respiration, due to water losses, for example. You also have to look at tomato, potato, cassava as organs that might also lose water and therefore have impact on their quality. Very interestingly, of course, if you go a bit deeper into this mechanism, you can also actually study what are the genes that are involved. Again, I will not go too much into details, but here you see that people have started to look at what are the genes that are involved in this mechanism. How do the plant is actually biosynthesizing those polymers in order to prevent or to mount this barrier directly at the wound site. And what you see here are some construction, transgenic tomato or potato, with a promoter region of a gene that is involved in this mechanism. And what we see is that this gene becomes very active upon wounding. So it means that upon wounding, the plant or the cells that are present there in the tuber or in the fruit are basically starting to synthesize some enzymes that are able to make those biopolymers or to make accumulation, trigger accumulation of those biopolymers, in this case, suberin. So our objective here is to study a similar mechanism in cassava. I should say that cassava is actually a very poor suberized crop, or at least very poor at suberization upon wounding. So the problem you have with this is not only that, of course, it represents an entry point for pathogens or water losses, but also cassava, as I said before, is suffering from the so-called post-harvest physiological deterioration, the so-called PPD. This is what you see here. It means that when you harvest cassava, and of course it is very difficult to harvest cassava without removing the roots from the stem, without damaging the roots, because you have to see the roots are quite long and quite deep in the ground. So very often they get broken during harvest. So it means you are creating an entry point for oxygen in those plants. The only way to prevent what you see here on the slide, after 7 days, you see this decay that is coming into the plant with the accumulation of different molecules that provide, or that gives a very bad taste, actually, a very bad property to the root. The only way to do this is to coat, to wax the root. And this is what I explained before, what people would do when they want to export to Europe or to America. So for markets where there is actually sufficient benefit or added value so that you can justify the cost of coating. But of course, in practice, if you use cassava as a food security crop or as a small cash crop, of course, you don't have this option. So we need to find different options. We've published different, well, summarizing all the different options. As I said before, many options are very difficult to implement for small-scale farmers. So, of course, one of the ideas is to look at the diversity of cassava and to look whether in this diversity of cassava, do we have variation in the capacity to heal? Again, I will not go too much into the details, but what you see here is that we have taken cassava cultivars, genotypes, varieties, you name it as you want, from the African context. And we've looked now whether they have different accumulation level of those biopolymers. Do they have a different capacity to biosynthesize those polymers? That's something quite novel, actually, because if you have to think that in terms of breeding efforts we have done over the last century for food security crop, but also for commodities, also in Europe, the major focus has always been to produce. So yield is the main focus for breeding. We want to produce more tons per hectare. That's been a major focus over the last decades or century, I should say. So it means it's a kind of a new paradigm. You need to look now at the diversity from a different angle. It's not only about yield, but it's also about capacity to have a long or prolonged shelf life. In this case, we look now at the total suberin, and we see that there is variation between those different genotypes that are being used by farmers, in this case, in Kenya. And we can also, to add a layer of complexity, you might also look at the composition of the suberin, because it might also be that the biopolymers is not only about, that diversity is not only about the quantity between different genetics, different genotypes, but it might also be in the quality or in the properties of this suberin, in its capacity to form a proper barrier for oxygen or pathogens or whatever. So this is one of the work we've done, quite pioneering work, because nobody has really looked into those properties so far, at least for cassava. As I said before, potato is a better example in terms of fundamental studies, because it is being used as a model species actually to study those mechanisms. Okay, so what you see here is indeed that composition might also vary between the different genotypes. So we do what you see here. We see hyper accumulator, moderate accumulator of suberin, and low accumulators. And we also see that there is some kind of variation also in terms of composition. Again, I will not go into the details, but important for you to have this in mind. It means that you might be also developing some kind of a fingerprint of those composition, while accumulation and quality, so quantity-wise and quality-wise, you might be also able to discriminate those different genetics. And it might be in the future possible to actually, if we demonstrate that this is important for healing, we might also be able to use this as a marker for breeding. You know, that marker-assisted breeding, so being able to identify the good parents, the one having the right properties, is also important. So this is what we are developing in this project, trying to have a fingerprint of what is happening in the plant, what are the properties regarding suberin composition and accumulation. So we see, of course, we're trying to also correlate this suberin composition with some tests on PPD. So we have also developed very standard and high throughput tests for PPD susceptibility or resistance. And indeed, we are trying to see whether some of those suberin composition parameters do correlate with this PPD mechanism. So we have identified what you see here. We are basically measuring also not only the initial composition of suberin, but also the capacity to biosynthesize. If you think about damage, wounding, of course, you are creating a new, you expose new cells to the environment, and therefore those cells need to also have the capacity. So you might accumulate good level of suberin in your periosteum, but that might not be sufficient. You might also need to have a capacity to biosynthesize this upon wounding. So this is another element that we are studying in our system. I showed you before that using quite elaborated biotech approaches, we are also able to monitor which genes are being expressed upon wounding, for example. So what we are also doing, taking advantage of fundamental knowledge created or generated in other model species, is trying to identify what we call transcription factors in order to see whether some of them might be involved in this biosynthesis of suberin polymers. This is quite interesting, the concept of transcription factors, because you might think that one single gene might not be sufficient to create this new phenotype, this new capacity to biosynthesize suberin. So in this case, using transcription factors or trying to study their effect in the plant allows us to consider deregulating or regulating multiple genes at the same time. So those transcription factors are basically sitting at the node, at the core of a control center, let's say, that is controlling many different genes involved in such mechanism. So that's also another interesting approach that we are trying to implement here. So we have, for example, here from RNA-seq data, again, I will not go into the details, we do find some of those transcription factors that we know are potentially involved in regulating genes of the pathway, of suberin pathway, that are deregulated upon wounding. So we test now whether they use or whether they create change in the expression. Again, I won't go into the details because I see I don't have much time left, but what we are demonstrating is that some of those transcription factors from cassava are able to deregulate or to change the expression level of certain genes in the plant. In this case, we use a tobacco plant as a model species for studying this. And then, well, last but not least, what you see here indeed is the composition or the accumulation of suberin in cassava and potato, a comparison. So what we see is indeed that potato is a much better crop or a much better species to synthesize and accumulate suberin. So this is what you see here in quantity. You also see that the quality or the proportion of the different lengths of those polymers do vary between the two species, potato and cassava. So we're also interested to see whether this translates into different properties in terms of blocking oxygen or water losses in the plant. So we have some elegant system here. What you see, we are taking the peridot and we are also able to measure exchange of water outside and inside this small compartment. And we are measuring, basically, we are looking whether when we remove those waxes, when we remove this suberin, whether we do change the permeance of the peridot. And this is what you see here. You see that there's quite some difference indeed that those molecules, those polymers do perform well in preventing water losses in this case. And we see that there's also some kind of difference between potato and two cassava genotypes that have been selected here that you see here. However, we consider that this is kind of similar level of transpiration. So at least initially, there is not a major difference between the peridot from the potato and from the cassava. We believe that the difference comes more from the capacity to biosynthesize upon wounding. We do different types of experiments. What you see here is also further investigation to demonstrate that actually in the PPD mechanism, it's not only the oxygen that do enter from the wound side that is important, but we do see that there's also exchange or something coming from the peridot, from the full root, that is also participating in this PPD process. So we see that it's also very important to understand the dynamics of exchange at the peridot level to explain how this PPD is taking place in cassava. I'm coming to an end. I said before that we were also interested in looking, well, we see that this work of investigating the pathway of wound healing, suberin biosynthesis in cassava is a very promising way, but it's a long way. It's a long shot if we think in terms of genetics, because it means first to characterize, identify the genes that are relevant for this mechanism, and only then starting to have a breeding scheme to bring those properties into improve cultivars for farmers. So one of the things we have also been doing at the same time while we're establishing methods to extract this suberin from potato in cassava is to also think, can those byproducts from processing of cassava, processing of potato, can we have some kind of valorization? And in this case, what you see here is basically extraction of suberin from potato or from cassava peels, and we use those extracts to actually develop a coating. And you see here that using coatings from cassava or potato, we are also able to reduce decay from fruits. In this case, we use berries or small tomatoes, cherry tomatoes. And we see that by coating those fruits with the cassava suberin, we are also able to reduce water losses in those fruits. It's quite interesting because, of course, in the African context, cassava becoming more and more important. I didn't mention this, but high quality starch that can compete with maize or potato starch because of purity, because of other properties. So there is a very strong incentive to develop a value chain for cassava production and processing in Africa with the development of many different plants in different African countries. And therefore, they generate a lot of byproducts from producing cassava flour because they do process those roots. And one of the byproducts is, of course, the peel. They need to remove the peel for processing the cassava roots. So there is well, usually people valorize this as animal feed. That's one of the way it is valorized, but actually that's quite a low valorization potential, at least in terms of income. So if we could develop also next generation chemistry for such a byproduct, by basically valorizing the suberin that is present in those byproducts, that would be a major advantage. So you would see here that we also gain the capacity on the African continent to also start to preserve other crops, other food production by using those byproducts from cassava production. I will skip this. I would like to thank you. I hope I was not too long. I was rushing, I'm aware, so you might have some questions. You can, of course, always contact me outside of this session if you have more questions. You see here all the people involved. You see the trip we had to Kenya to meet with some farmers, some women association of cassava production, but also visiting the memorial of Mandela actually in South Africa as we were nearby when we were visiting all the factories for food processing and seed processing in South Africa in Pietermaritzburg. Thank you again for listening to my talk.
Thank you, Professor van der Schuren. That was very interesting talk about the deterioration of cassava. We have a few questions here from the audience, and one says, agricultural losses is very high in rural areas and refugee settlements, mostly in the rainy season. Where here in Africa, Uganda. How these skills can be extended to refugee settings? I imagine like they mean your research.
Well, I'm not sure we research on this. Of course, I didn't mention, but since you are talking about human areas, of course, we also do see a major impact of climate change on the post-harvest losses. That would also be a way to mitigate, of course, with our research. I think people have to be aware that, as I said before, because of and it was justified when we wanted to increase food security to take the angle of yields improvement or yield increase. There is more than to this, and I think what people have to realize, well, of course, we would like to contribute to food security in refugee camps, for example, or in any other context where it's very important. This is it's a long process because now we are discovering this diversity, but by the time we are able to mobilize this new knowledge into practical solutions for farmers, that will take some time. But I think it's very important to start. I should also mention that cassava is still considered as an orphan crop. So it means also that the industry is not so much investing as we don't have the investment we do have in maize, corn, potato in terms of research and development of products, of varieties, simply because cassava farmers cannot really afford to buy expensive seeds, for example. So that's also something that we need to contextualize. That's also why capacity building, which we also have done during this project, to basically empower this type of research in African universities is extremely important, because for some of the crops, not only cassava, and we can name other crops in Africa, teff, sorghum, whatever, it's also very important that local research has to be developed, and it has to be somehow cutting edge as well to develop novel solutions.
Okay, perfect. We have a second question here. Somebody says, thank you very much for your nice presentation. Cassava is really an important food crop in Ghana. It's mainly used to make gari, powdered cassava, to make fufu, powdered cassava. I wanted to know if your study could be applied to Ghana as well, since there are different species and the resistance to decay. You mentioned most farmers use wax to protect the cassava from post-harvest. Currently, what do you suggest are the best ways to protect our cassava after post-harvest? Thank you.
Yeah, it's a very big challenge. Actually, Ghana is indeed a big producer, as Nigeria, of cassava. They do locally process. Also, Ivory Coast would be another one to mention with Atieke. However, actually, I had the chance to visit the Nestle factory in Abidjan, where they actually source high-quality cassava, or high-quality starch, I should say, from Ghana, actually. And that's a big challenge for them, because indeed this transportation from Ghana into Ivory Coast takes several days, and by that time cassava is actually decaying. So indeed they're looking for solutions, but there is none, because the problem is that you cannot manipulate or process the cassava in the field there in Ghana or elsewhere, but that's a big potential for them. One solution that's been implemented over the last decade is the so-called AMPU solution. So it means autonomous mobile processing factories, that actually units that are actually moving through villages, and where they basically process directly cassava into some kind of dough that can then be exported to the factory for further processing. So this, I found a quite elegant way, still very difficult, because you might think that in terms of transportation, those big trucks with the kind of a small processing factory are difficult, or it is challenging to bring them close to the farmers. However, this is one of the practical solutions that has been implemented. But for sure, if we could develop varieties that can have reduced losses in terms of PPD, as I mentioned before, that would be a major gain for farmers. We estimate for example, that cassava roots on the Nairobi market, there have been some studies on that showing that after 3-4 days, the value is 1/5 of what it was initially. So after at the fresh, very fresh, you get a price, and 4 days later, it's only 1/5 of this price. So you can see the loss for the farmer also from a financial point of view as well for those small scale farmers.
Okay, yeah. Yeah, I guess there are like many solutions that needed to be like implemented, like potential solutions.
Yes.
I'm just gonna take this last one here. Somebody's asking is if the paraffin wax that is used to coat the cassava is it edible?
Well, people will not eat it, right? Because they will
Peel it
remove
off.
the peel. Nobody's eating the cassava with the peel. So
But it's no
this.
harm, as in if there's a little
That's
bit left.
no, 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 of course not.
Ah,
And in this case here, I mean, if you think we are all eating potato peels in some cases, not always, but I mean, and there is
mm-hmm.
full of sugar in there. So you can imagine that those kind of
Mm,
molecules are not, of course, not toxic for
yeah.
humans.
Perfect. Okay, Professor van der Schuren, thank you very much for sharing your research. Now we're going to move to Professor Pleisner. Professor Daniel Pleisner is Scientific Director at the Institute of Food and Environmental Research, ILU, in Germany. His areas of interest include biochemical and industrial engineering. He works in the area of marine biology and the evaluation of fermentation processes using microalgae to valorize different waste streams. He specializes in developing processes for the biological recycling of organic waste and conducting life cycle economic analysis of bioprocesses. He has extensive experience in coordination and management of projects in bioprocess engineering in Germany, Poland, Denmark, and Hong Kong. And the title of his talk today is Decentralized Bioprocesses for the Conversion of Residues with Low Climate Impact, which is part of the Climaqua project from the FOSC and ANET. Professor Pleisner, thank you very much for joining us today. I'm just gonna stop real quick to stop sharing my screen so you can start sharing your presentation. Thank you very much for joining us.
Thank you, Evelyn, and thank you for inviting me. So now I have to share. It should work.
Fingers crossed. Yeah.
Perfect.
Perfect. Now it's in the last layer. Yeah.
So here we are. So yeah, thank you again. It's a pleasure to talk today. Yeah, our project Climb Aqua, it comes to an end, and we have been dealing actually with the development of decentralized bioprocesses for the conversion of residues, and one big goal was to have a very low climate impact. I have to say we learned quite a lot in our Climb Aqua project, and I hope I can transfer some of the knowledge we gained within 15 minutes. Okay, how did it start, the whole project? So we were actually thinking, how can we combine the knowledge and the experience we have made in Norway and in Germany and the knowledge and experiences made in South Africa and Kenya to make something better, to develop something better in terms of utilization of waste and residues and so on. And we were thinking, okay, we have in Norway and we have in Germany, we have a very good waste management system. So we know how to collect organic materials, organic waste streams, residues, etc. And actually we know how to deal it. But we need more insight in what is the best process actually to convert our waste streams, and we need more input in what is actually the best product we have to produce from the waste stream. And then we talked to our partners in Africa and South Africa and in Kenya, and then okay, they have the issues of a climate change already for more years than we experiencing it actually in Europe. So they know how to deal with all the problems associated to climate change, but they have different conditions. Of course, they have more sunlight than we have in Europe, higher temperatures. So finally, it was a long discussion, but finally we ended up with, okay, what would be the best process for the utilization of waste streams using microalgae? Is it an autotrophic process or is it an heterotrophic process? So that was the final conclusion. And okay, let's go ahead with a joint project. And I have only a couple of minutes today, so I will only focus on the part I did in together with the partners in Norway. So I exclude the African part for now because it would be too much. So here you can see our approach. So we focused on residues from the fish processing or from aquaculture. And I have to minimize you, otherwise I can't see the slides. So it's, turn my pointer, hang on. It starts with the processing of fish. Then we get some fish parts, skins, bones, whatever, heads, which are not really usable for human consumption. So usually in a fish processing unit in Norway, those residues are hydrolyzed. They produce some protein hydrolysate from it and they sell it. It's a very nice product. They also produce some oil from it, but there is something left, and this is what you see here. It's I mean, we call it pellet. Here it's it does not really look very nice. It's not something you would give directly to humans to eat. So it basically is thrown away or ends up in a biogas plant. And we were looking for a different way of using it. And we used it as a nitrogen source for our cultivation of microalgae. And we also used wastewater from fish processing. And we used wastewater from the aquaculture sector. So it's basically slime collected in fish ponds. And here, I can go just one slide more. So we have 3 residues tested: wastewater, residues obtained after fish hydrolysis, and slime collected from the fish ponds. And we have 3 different residues or resources, but 2 of them are waste streams and 1 can be called actually as a byproduct. So the byproduct is the residues after fish hydrolysis, but wastewater and slime, that's waste. And I think this is something we did not really consider at the beginning. Of course, we were actually aware of the issues when we convert a waste stream into food and feed. And our intention is always to use the algal biomass as a food or a feed for whatever. So I had quite a lot of discussion with our national contact point, and they actually told me very clear that they don't want to be connected to a project that converts a waste stream into food and feed. I mean, they made it really clear. And the reason is the European Union actually excludes any conversion processes waste into food. They don't want to be connected to it. It's totally against regulations. For good reason, But this is actually something that's a tough statement we did not really consider at the beginning of the project, but we learned from it in the middle of the project. And we considered it in our reports and in publications, of course, but we also work now on that the European Union has to open its mind to allow the conversion of selected waste streams into food and feed. What can be used for sure? This is the pellet after fish hydrolysis, because this is not a waste stream. This is really a side stream, and this is not associated with any contaminations, microbial or chemical. But yeah, I can imagine wastewater and slime could be critical. Anyway, we used all 3 substrates in our project just to see what is possible and where are the drawbacks. And I have to mention we used for our cultivation Galieria sulfuraria, and I like Galieria sulfuraria very much. Galleria, first of all, grows at a pH 2 and at 45 degrees, so it needs higher temperatures. But Galleria grows in presence of organic waste streams. You can see here a nice picture where it's showing food waste that was hydrolyzed, and the hydrolysate used as a nutrient source for Galleria has been, I mean, everything started in 2017, not so long time ago. And luckily, there are specific strains that even accumulate phycocyanin under heterotrophic conditions. So there's no light involved, what makes the production fairly simple. Well, at least we expected that it is not a big deal to grow Galleria, but we found out it's actually not so easy as it seems. We promised in our Climb Aqua project that we have a non-sterile cultivation. We expected the pH 2 and the higher temperature can exclude most of the contaminants. It can exclude some contaminants, but unfortunately not all. So we have from time to time issues with some nasty fungi, which can also grow at a very low pH and higher temperature. But luckily, my team actually found a way to exclude the fungi, and if everything is working well, we can actually produce Galleria even under non-sterile conditions. But it took us some months to figure out how to do it. And here you can see our friends from the German Institute for Food Technology in Quakenbrück, DIL, probably you have heard from them. They constructed a pilot, not a pilot scale, it's a technical scale bioreactors, 200 liters in scale. It's a very simple system. It has been built for our project, actually. It has a stirrer, it has a pump, so we can do the hydrolysis in it, and we do the cultivation of the microalgae in it. You can see there's no light involved, it's just a vessel. And this was the starting point. And based on this reactor, we did all the calculations for the life cycle assessment, which I will show at the very last slides. But it was a long way. I must say it was a really long way from the starting of the project until we got this bioreactor done and the cultivation working in it. Growth performance. So just we have learned about the regulation. That's very important to consider. We have learned how important it is to really select waste or to separate waste from side stream. Side stream can be used. Waste streams cannot be used for food and feed. But we also have learned quite a lot about the effects of different substrates. For Galleria, we have tested so far quite a lot of substrates: digestate, starch processing water, food waste, straw hydrolysate, fish residues hydrolysate, and so on, wastewater, of course. So, and sometimes we have seen that Galleria grows very well, and sometimes we have seen that Galleria does not grow at all. But you cannot really know it from the beginning. You have to figure it out. This is why we always have to test the different substrates and media. Here you can see at the open dot. It stands for the ordinary medium. It's the control medium, so Galleria is supposed to grow in it. And here is a mixture of medium and wastewater. You can see there's actually no difference in the performance between the two mediums. So I can, okay, medium and wastewater is quite comparable. So we can actually use wastewater in our cultivation. And now the most important work could start. We tested the fish, we hydrolyzed once more the residues we got after fish hydrolysis to extract more nitrogen. I forgot to mention the FAN stands for the free amino nitrogen. It's actually amino acids, more or less. So this is the limiting factor actually in our cultivations. We need nitrogen, quite a lot of nitrogen. And sometimes we have from the residues, agriculture residues or wherever, not so much nitrogen available to obtain high cell densities. But for the hydrolyzed fish residues, we were lucky enough to get sufficient nitrogen out of it. So we tested at 20%, so it's a diluted pellet extract, and we did the hydrolyzed it once more, since for the 20% pellet extract hydrolyzed, and we tested a 40% pellet extract. And you can see here the 4 different media actually behave differently. So we tested wastewater once more. You can see here as a pure wastewater without medium. So the best growth has been seen in the 20% hydrolyzed pellet extract. You can see that it did not stop growing even after 6 days. So in the 20% pellet extract, which was not hydrolyzed by us, growth stopped after 5 days, and then the cell number declined. And there was no growth at all in the 40% pellet extract. So it was just, okay, no growth at all is wrong, but just very little. But anyway, was not much happening. You can see also the glucose concentration in all flasks where Garderia was growing. We could see a consumption of glucose where Garderia was not growing, glucose remained stable. So this is something we noticed actually, that when we can measure different things. We can measure the concentration of glucose. We can measure the concentration of long and short fatty acids, and we can measure the concentration of nitrogen and of phosphate, and so on and so on and so on. But here in this particular medium, there's something in the background that inhibits the growth of Garderia. And you can see here, first we expected it might be the concentration of amino acids, but this is not true, because here where actually Garderia did grow very well, in the 20% hydrolyzed pellet extract, we had 1.4 grams per liter FAN. And here where Garderia did not grow at all, we had something about 0.6. Then it fluctuated a bit because there was more of the proteins hydrolyzed during the cultivation actually. So not much happened. So it might be related that the protein concentration in the medium was simply too high for Garderia, that there was an inhibition, or there might be also associated with the fats or whatever. So this is what we have learned. It's never as easy as one may expect at the beginning of a project. We have really carefully select the different substrates, and we have to test it always. Yeah, growth rate is very important. You can see here when Garderia did grow, the growth rate was very well in the control medium 0.79, and the best growth rate we got in the 20% pellet extract was 0.92, but the highest concentration was obtained in the 20% hydrolyzed pellet extract. So I was very lucky actually with the numbers, but I was hoping for that we can use a more concentrated pellet extract for our experiments, because then we can also add more nitrogen and don't have to feed from time to time, and we can just keep the system closed and have reduced risk of contaminations. I mean, when we open it and feed and then close it again, there's always a risk that we introduce something to the system which we don't want to have. And just as a reminder, we did everything non-sterile. Yeah, assessment of climate impact. I mean, and this was done in flask cultures, and we took the best information and did the cultivation in our demonstration plant at 200 liter scale. And we had to collect many information in order to assess the climate impact. So it was what we did not stop actually at bioprocess. So our boundaries was very wide, so to say. We started at the fish harvesting of live fish, and the fish processing was included in the assessment, and hydrolysis of fish waste, and the cultivation of microalgae, the harvesting of microalgae, and the feed preparation. And also, we included the whole chain, so to say. So we want to make it real and as realistic as possible, and not just look at the cultivation process. Then I just show you a simplified figure here. This is all the millipoints. So I don't know if you know this, but 1 point stands for the environmental load of one average citizen in the European Union. So, and this is millipoints. You can see here different examples. So the Calderia biomass, beef meat, consumption egg, soybean protein concentrate, soybean protein isolate, pig meat, and chicken meat. So all the different protein sources we have around. And the blue column here, this is Calderia. Calderia, 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 and Calderia, and all the others. So let's compare it first with beef meat. Or what is associated to human health. So beef is actually bad, and Calderia production would be much better. The same applies for the ecosystem quality, for the climate change, and for the resources. In some cases, I mean, pig meat production is from the human health perspective better than our Calderia production. It's also, stupid. Not pig meat, chicken meat production is better in terms of human health than Calderia. It's also better in terms of ecosystem quality, and also better for the climate change, and also better in terms of resources. So this is a realistic life cycle assessment, including the whole chain. I mean, we can also cheat a little bit and make the boundaries more narrow. Then we probably would have perfect timing. We have in Germany a test alarm, so all mobiles ring at the same time. So that's the reason why. So yes, we have to optimize our process, that's for sure, in order to get a much better climate impact. And we have to put different processes in the perspective of the climate. So, and perhaps I will give another presentation very soon where I can show the results from Africa, because then we talk about the autotrophic cultivation of chlorella and Calderia. And then the final task would be to select the conditions and to select where can we produce, and actually to select how can we produce where. So this would be the correct term. Okay, to conclude, I mean, we have learned quite a lot. We have a lot of byproducts available from the aquaculture sector and also from the agriculture sector, and we should keep all resources in mind. We should not exclude waste streams. We should include all for a sustainable bioeconomy. And but what we have to keep in mind in order to proceed with a change in regulation, that we should be aware that we not recycle or not circulate all the good nitrogens actually, but also all the bad things that are present, heavy metals, pesticides, and so on can be present. No need, but they can be present, and we have to just to figure it out if we have something present, and if not, then we can just go ahead and produce food and feed from all materials. Yeah, that was a quick presentations, and thank you very much for your attention. And if you have a question, just drop me a mail or give me a call. And you can also check our homepage. We have quite of interesting projects going on. And if you would like to cooperate or to join the project, let me know. Thank you very much.
Thank you very much, Professor Pleisner. That was a very interesting presentation. And somebody was asking about your email address, so you had it there. So I presume they took note of your address. We have a question here. Somebody saying a very interesting application. My question is, how to use the same nutrients for the growth of Galideria, considering use a wide variety waste from restaurants and bakeries. I imagine kind of a different type of waste stream.
Oh, that's a big issue, I have to say, and we are on it. I got this type of questions actually every time. So we we have one big issue. We have no quality management when it comes to the substrate. We cannot actually, for the moment, there's no tool available that gives you any real-time information about the quality and the quantity of nutrients in the waste stream. It depends when you collect it, where you collect it. The season of the year is important also. So they have a fluctuation in quantity and quality. So yeah, we try a new project actually where we measure in real time the composition of the waste. And then depending on the composition of the waste, we will define a pre-treatment, first of all, to get the nutrients out, which we need for Galdieria, sugars, amino acids, phosphate. I didn't mention phosphate at all, but phosphate is also important. And yeah, if we measure that we cannot use the substrate for Galdieria, we can give it to another bioprocess. It does not mean that we have to produce Galdieria. We can also produce something else.
Mm-hmm.
But it's a good question and it's a tough issue, and I have no answer right now how to do it.
Yeah, sometimes it's really hard to like answer. Yeah, it's just it's like the experimentation and the project is ongoing. So I have a question though. So I saw in your research that you started working with Arthrospira platensis, and then you moved to Galideria. Like was the main issue with Arthrospira? It was not growing as you were expecting, or?
It was not growing as we were expected.
Mm.
Exactly. That's the point. We I mean, we have two different aspects. People in South Africa and Kenya do the cultivation autotrophically. So they have no issue in cultivating Atrospira. But we could not do it heterotrophically. We have seen a growth, but it was not, I mean, it was so slow.
Oh, okay.
It didn't make any sense to go ahead with it because we had to produce a certain amount of feed for feeding trials in Norway. They fed salmons with it, and we figured out, luckily at the very beginning of the project, okay, we have a big issue here, so we have to change the strain, and then we decided to go for Galdieria.
Ah, okay. And then did you test different like algae and before you settled for Galideria, or you kind of knew that that one was the other option?
We did not test different algae. We just selected Galdieria because we had, first of all, experiences with the heterotrophic growth, and we did know that it can grow in different waste streams. And so okay, let's be on the safe side and let's take Galdieria for it.
And the fact that it's like a extremophile that it grows in pH 2, is that better or it is restricted?
It's Atrospira was supposed to grow at a pH 10. That was the starting point for the non-sterile cultivation, and we had to select an extremophilic strain as well. So that is why there's also good reason why we chose the Galdieria. It grows at pH 2. So and pH 2 is good, but causes some other troubles as well. So corrosion of materials, etc., etc.
Mm-hmm.
Yeah.
Okay. That's very interesting. So I think I'm gonna leave it at that because we're past 10. So Thank you very much, Professor van der Schuren and Professor Pleisner, for joining us today and for sharing all your expertise. And thank you everyone for your questions. Unfortunately, I won't be able to take more questions today, so I can let everyone go. So thank you very much for joining today, and I see you in another opportunity.
Thank you very much.
Bye
Thank you very
bye.
much.
Thank
Bye-bye.
you.